Hello everyone. So today I'm going to start biology chapter 2, that is sexual reproduction in flowering plants of NCERT textbook. In the previous chapters we have got about different stages and events of sexual reproduction in plants. That is the events of sexual reproduction, which is the pre-fertilization event, fertilization event and post-fertilization event. In this chapter, we will be dealing with elaborate description of all those events, including the morphological structures and structures of the reproductive organs and how the gamete formation takes place, what happens during fertilization, and yes, of course, about the post-fertilization events in flowering plants. In this part of the video, I'm going to discuss about the morphological structure of a flower and microsporogenesis taking place in flowering plants. Microsporogenesis means how formation of male gamete takes place in the flowering plants. So let's start. Okay, whenever we see a flower always attracts us by its scent or by its attractive color. In human rituals, or you can say in our human life, we use flowers for different purposes, whether it's be a cultural function or whether it be a token of love or whether it be in some mourning. We always have been using flowers, but to a biologist or scientifically, a flower has a different value. It is the reproductive part of a flower that helps in continuity of the species of the plant. So let's discuss the different parts of a flower. Looking at the petals or sepals, we always used to say that they are the attractive parts of a flower. They used to attract insect, or they used to attract human beings, or they used to attract other animals. But scientifically, these petals and sepals have their own purposes. So let us see the importance of different parts of the flower. Okay, so first of all, we are going to start with the receptacle. Receptacle is the part that used to hold the flower. You can see in the diagram the receptacle is a swollen portion. Actually, the receptacle always used to have a swollen portion because the flower is too big for the pedicel to hold it. That's why the receptacle, being broader in size, helps in holding the flower in its position without breaking. Then comes your sepals. Sepals are mostly green in colors except the perianth, whose sepals and petals are of same color as you have got in class 11. So sepals used to cover the flower during the bud formation and helps in protecting the outer layer of the flower, or you can say help as a protective outer layer that helps in protection of the reproductive parts of the flower. As the flower slowly matures, it's used to spread out the colorful petals. These petals, or we can also say corolla, are colorful or having scent. These also have some scientific purposes. The color and the perfume or scent which are being spread by the flower used to attract insects or other animals for pollination. So these parts which do not involve directly in reproduction, that is the sepals or petals, we call them as the accessory part of the flower. Whereas the male gametes and the female gametes formation takes place in anther and your ovary respectively. These are called the female parts of the flower. So the androecium or stamen is said to be the male part, and the carpel, pistil, or gynoecium is said to be the female part of the flower. So one by one, let's go to the detail of the reproductive structure of a flower. We are going to start with the structure of androecium, that is the male reproductive organ, and what is microsporangium. But before that, I would like to say something about the flowers. We know that a flower blooms, but the destination of a flower, or that a flower is going to bloom, has been decided a very long time back. The decision that the plant is going to flower, or the primordium, that means the apex portion, instead of blooming leaves is going to bloom flower over there, has been taken place since long time back. Several hormones and structural changes are initiated, which leads to the differentiation and further development of the floral primordium. Inflorescences are formed, which bears the floral buds and then the flowers. In the flowers, the male and female reproductive structures, the androecium and the gynoecium, differentiates, and slowly and gradually they develops into mature male and female reproductive organs, releasing male gamete and female gametes. So, talking about the androecium, we know that the androecium or stamen is made up of two parts. The anther, you can see lobular structures over there, and a long stalk known as the filament. The proximal end of the filament are mostly attached to the thalamus, or the, you can say, end part of the petals. This stalk or filament used to vary in size and length in different flowers. If you see different species or different floral species, you will see that the structure of the anther varies in all of them. The diagram shown here shows you a mature pollen grain where the pollen grains are being dehiscensed, or they have been burst out. But before that, we have to learn that how this pollen grain formation has taken place, or what is pollen grain. As in the diagram, you can see that the anther are shown with two lobules like structure formation over there. Most probably, an androecium anther are usually bilobed in structure, having two lobules formation in them. Each lobule has got two sac-like structures in them that divides them into two thecas. So we call them as dithecous. Most of the androecium are dithecous in structures. Each lobule have got two theca, and all the androecium, or you can say all the flowering plants, used to have bilobed anther. That means two theca in each lobe, consisting of four thecas together. This diagram is showing the transverse section of an anther, where you can see the pollen sac, or you can say a sac-like structures present in them. These sac-like structures are the structures where pollen formation or male gamete formation takes place. That's why we call the sac-like structures as microsporangium. The term micro is generally used for male reproductive organs or male gamete formation, whereas the term mega is generally related with the formation or structure of the female reproductive structures, as the male reproductive structures and the male gametes are usually smaller in size than the female gametes. So, the sac-like structures where male gametes are formed is termed as microsporangium. So, let us have a look at how the pollen grains or the male gamete formation takes place. As I have already said that the process of formation of male gametes in anther is known as microsporogenesis. Before going through the process of the formation of male gametes, let us have a detailed view of the transverse section of anther once again. The first picture depicts the transverse section of a young anther showing the four thecas, bilobe structure of an anther, and connective, which is the group of vascular bundles connecting both the bilobe structure of the anther together. Here, the next diagram shows us an enlarged view of the microsporangium, which is showing us the sac-like structures in between, or the cellular structures over there are the microsporogenous cells or microspore cells, which are going to form future pollen grains. The outer layer just after the microsporangium is known as the tapetum layer. Tapetum layer acts as a nurse cell, nourishing the sporogenous tissues or sporogenous cell so that they are able to derive nutrition from them and grow as male gamete. After the tapetum, there are 3 to 4 layers of cells known as middle layer, which helps in protecting the sporangium from outer injury. The last layer or the outermost layer is the epidermis or endothecium that helps as an outer covering of the anther and helps in protecting the anther from outer injuries. Now let's move on to the process of microsporogenesis. The process of microsporogenesis takes place in four sac-like structures of the anther, that is the microsporangium. Each microsporangium consists of a group of cells known as sporogenous tissues. The sporogenous tissues used to gather nutrition from the tapetum layer, and each sporogenous cells divide themselves or undergo reductional division to form a tetrad. The cells which are capable of undergoing reductional division are called microspore mother cell or pollen mother cell, also designated as PMC. The pollen mother cell is the cell that is capable of undergoing reductional division to form a microspore tetrad. In the next diagram depicted below, you will be able to see the formation of tetrads from the sporogenous cells. These tetrads, on maturing, undergo pollen formation. Each tetrads give rise to 4 pollen grains. So if we want to see the whole process of microsporogenesis, we will be able to see that in microsporogenesis, the sporogenous tissues or microsporangium, which are twice in number, each cell act as a pollen mother cell or microspore mother cell, which are twice n in nature. This pollen mother cell undergo meiotic division to form microspore tetrad. The pollen mother cell are also known as meiocyte because they undergo reductional division to form microspore tetrad, which are haploid in nature. Each cell of the microspore tetrad starts releasing a protein known as sporopollenin, which is the hardest known substance in the biological world. The sporopollenin forms an inner layer around the cell known as intine and an outer layer known as the exine to form the pollen grain. Let's see a detailed study of the formation of pollen grain. In this diagram, you'll be able to see how each cell germinates into the formation of bigger size cells that further divides into a large vegetative cells and a small generative cell. The generative cell gives rise to the male gamete formation, whereas the vegetative cell helps in the formation of pollen tube so that the male gamete can easily reach the female gamete without any damage. There is a sectional view of the mature pollen grain showing the different layers of pollen. You'll be able to see the outer exine layer formation and the inner intine layer formation. Out of the exine layer formation, there are some portions which are left without the formation of exine layer. This is known as the germ pore. The germ pore is the portion from where pollen tube formation takes place. As I have already said that sporopollenin is the hardest known substance in the biological world, so it is difficult to break the substance. So at the germ pore, the layer of exine is not present so that it is easy for the pollen tube formation to take place. With the formation of the pollen grain, the process of microgenesis ends, and that goes the next process, that is transfer of pollen grain from male part or male reproductive part into the female part. This is generally called pollination. For pollination to take place, dehiscence of the anther takes place first. The anther bursts its pollen grain out so that it can be carried by different pollinating agent from the male reproductive organ into the female. The next diagram shows the different structures of pollen grains. That shows that the exine and intern layer formation used to form different outer structure in the pollen grain, varying the shape, size, and structures of pollen grains in different species. So with this, the formation or process of microgenesis ends and the next process starts formation. And in the next video, we will be looking for how the process of megasporogenesis or female gamete formation takes place.